Okay, thank you all very much for coming. Thank you, Olga, for organizing. I have 50 minutes left, I see. I thought I had 30 minutes. Okay, I'm joking. I'm going to talk about, for the coming 50 minutes, about proteomics and how to create value out of data. But before I start, I would like to see a few hands. So I would like to ask you, which of you have a background in biochemistry? So that will be me. Okay, biochemistry background. And which of you have a background in data science or data analysis? Okay, so that's almost equal. And which of you would like to work with data science in the coming 5 years? Okay, that's quite even numbers. That's good. Okay, so yeah, maybe it's good to start a little bit where I come from. So I did a PhD in biochemistry, and my PhD was focused on one protein group, which were protein tyrosine phosphatases, and it was based on the standard paradigm of science, right? So most of it. So you either take a protein away and you see what happens, or you overexpress a protein, and then you see what happens. And so that's what I did, and it resulted in publications, which is very important also for my future. But what I always was thinking is that you kind of miss the big picture, right? Because I was looking at protein tyrosine phosphatase and the relationships in cancer, and I was thinking, you know, there's so many proteins involved in cancer, and I only look at protein tyrosine phosphatases. And therefore, I got a little bit frustrated. And if you look at cancer in general, it's such a complex disease that, from a disease point of view, there are many diseases like cancer. So looking at that, and okay, if you look at cancer, of course, it's not only that there are many types of cancer, but there are also differences between people. So anyway, I got a little bit frustrated, and I chose to focus on proteomics. So I decided to go to the UK, to Cambridge, and to not good. Okay, I won't move. To the UK. Oh, that's better. Yeah. And to focus on proteomics. And there were two things that immediately were difficult, right? So first of all, if you look at proteomics and you look at all those proteins, you get a result of 150 proteins or phosphopeptides involved in a certain disease. What are you going to do with this data? You can't go to a GP and say, look, this fantastic diagnostic assay based on 150 phosphopeptides. It just doesn't work. And the second problem that I got is that many data sets are actually flawed, because if you don't have the right controls, if you don't have the right experimental setup, it actually means that before you start your experimental experiment, it's already been flawed. So lots of data has been produced, or is being produced, but there's not much that can be done with it, because it's already flawed. Anyway, next slide. Do I have to walk there? Ah, I see. So this is proteomics in the way that, oh shit. So this is a fish, and this is the fishing experience. So if you focus on just one protein, right? This is proteomics, when you focus on many proteins at the same time. And when I talk about proteomics, it's not only about identification and quantitation, but it's also about protein-protein interactions, subcellular localization, and post-translational modifications. So there are many ways how you can look at proteomics, and I'm going to look at a few technologies that are around, that most of you probably will know, going from small scale to large scale, and then see what the problems are. So there's the classical Western blot that everyone knows, so you target the protein with an antibody. Then there is here an approach where you look at multiple proteins that can be with targeted proteomics, but also with protein arrays. And then here you have, say, the big data, where you look at thousands of peptides, protein spectra at the same time, and then you try to extract data of interest. So I'm going a little bit into more detail on all of that. So this is the classic Western blot. So it's cool because it works, of course. It's cheap and everyone knows how to do it. It's nice to interpret, but it's very labor intensive. I don't know if you've done Western blot, but after a few years, was totally fed up. And you can look at only one protein at the same time. If you go through this slide, which is much easier for me to explain after the other presentations, this is an approach where you look at multiple proteins at the same time, targeted proteomics. So how it works is that you select your peptide of interest, you fragment it, and you analyze it, in this case with an Orbitrap. And as you've heard before, the spectra over here, you see the difference between the peaks of the spectra is definitely the mass of an amino acid. So you can just read the mass from right to left. That's your identification. And the peak beneath a chromatogram is the quantitation. So in this case, you have identification and quantitation, and you could look at that for tens, hundreds of peptides, and you can actually still do it by hand. It's cumbersome, but you can still do it. But if you go to a real full proteomics approach, over here, which is a proteomics approach that we as ProteiQ use, it's called data-independent analysis. And what we do here is that you look over the complete range of M over Z. You look, you divide the complete range in this what we call swath, and every swath takes 64 milliseconds. So for a 40-minute run, that will mean that you have around 37,500 spectra. And this looks, a bit like this, or we have also a different way of showing this. So this is just, we zoomed in here, so here you have the M over Z, and here you have a peptide of interest, and you see already there's a huge complexity. And in order to know what it means, you need to have very good data science skills. But if you know this, and if you master this kind of work, then you can really answer really interesting questions that you could not do before. And therefore, I have two examples that show this. So this is an example of a study where they look at the effect of weight loss on your proteins in blood. So they looked at 53 obese individuals. They had quadruplicates in terms of numbers. Then they looked at them over time, and that resulted in over 1,200 different proteomes. So what I just showed was one proteome, so this is over 1,300 proteomes. So that's a lot of work, and therefore you need good data science skills. And it resulted in 93 significantly affected proteins. And there were many conclusions you could draw, but one of the conclusions was very important. And what therefore I've taken out is that the weight loss results in the decrease of CRP and CA1, which are two known markers for cardiovascular diseases. So they saw that if you lose weight, these two markers, which are correlated with cardiovascular disease, are downregulated. So you actually can use this now as a new diagnostic tool to monitor and optimize treatment of obese patients with high risk of CVD. So you immediately get something back out of this data. Another example is an example where it's really systems biology, so they don't look at only at the proteins, but they look at different kinds of data. So they used here environmental factors, and they used symptoms, and also protein-protein interaction network. And they used that information to predict the outcome. So they could stratify patients and predict which patients will be susceptible to a certain type of treatment, on which you can see here in the terms of an inhaler. So you could do many things with it, that's all great, but I think it's also important to know what the challenges are, because I'm a biochemist and I don't really have a data scientist background, but it's great and it's fun, but it's also complicated. And one of the things that are complicated is that the quality of the data you're working with is the quality of the data you're working with. Okay. Just say, if you can't hear me, just say something. The quality of the data you're working with can differ a lot. So you can have lots of bad data, or you can have lots of noise in your data. If you look at the results of genomics and proteomics, they do not always align. If you look at the data of a Western blot and the data of a mass spectrometer, even between vendors, it can differ. And therefore, there's a high complexity from that point of view, which you have to deal with. The second point is the complexity of modeling. So you need to find the best way to describe the relationships of information that are within your data. So there are choices you have to make here. Which kind of modeling route am I going to take? And of course, there's the always occurring false discovery rate, which means that if you look at large amounts of data, there will always be some parts of the data that is not good. In other words, the null hypothesis is rejected, but it should not have been rejected. Okay? So the more data you have, the more false discoveries you have, and that's something you have to think of. So if you make it too stringent, that means that you throw away a lot of data. If you make it too open, that means you have too many false data within your data set. Okay? So false discovery rate is actually tough, and there are many ways on how to deal with this. So these are the challenges. So I've been walking around in biochemistry and data science for quite a while, and I think one of the needs that I find that people should have, or in a perfect scenario, the needs that people should have when they work in this kind of environment, is that they have to be multidisciplinary. Okay. And this means that all these three things, which is sample preparation, okay, lab experience, how to run a MS, at least have the idea about the theory, and about the data analysis and have the skills to analyze data, these all should be all three should be one person. So obviously, if you have the data analysis skills, it's not enough. You have to know how mass spectrometry works, and preferably you also know how sample preparation works. And if you come from the other way around and you know all this, but you don't have the right skills, you also cannot go to the core of the problem. And of course, maybe many of you know this, but I want to stress it because it's so important. You need to ask yourself the good questions, because, like I said in the beginning, only data is not good. It's not enough. You need to know what you're looking for. You need a hypothesis. I once was in a conference, and somebody was talking, about her or his project. I can't remember anymore. And at the end of the project, somebody got up and he just said, "So what?" Okay, which is a bit impolite. But anyway, the good thing about the question is, and it makes you remember, it was a very good question. So why would you care? And I think it's very good to think before you start what actually are you looking for. And then the second one is the experimental design. It's very important to have, for example, the right controls and the right numbers. If I work with data scientists before, they know that a lot of biologists or people for medicine, they come to them and they say, "Look, this is my data. I want to publish it. Just analyze the data." And they can't, right? So they say, "Yeah, I mean, your experiment was dead from the beginning. Before you started, it was already dead. So you have wasted resources and you wasted money, and nothing I can do with it. You can't resurrect a dead person, a dead experiment." So think very carefully, and it's very hard. And of course, the last bit is you need to be open-minded when you look at data. So don't try to focus too much on what you want to publish, although it's hard. Look at it. What's your quality? Be open-minded. That you just be open-minded. I think that's the core. And then of course, it's a bit fallen off, but you need to develop skills. You need to develop skills and algorithms. There are many algorithms around already. It's not something that you have to do anything from scratch, but they are around. You have to understand them. You have to work with them. So from that point of view, it can be quite a hard bit. That sounds a bit negative, so I would like to end with something positive. I think when everything comes together, and you want to go into data science, I think there's a tremendous future for people in data science, because there's so much work going on and so much interesting things will be discovered. And the opportunities that will come around for data scientists in order to add value to biology, to medicine, society in general, will absolutely be enormous. So incredible! I'm in time, it seems, because I haven't seen Olga saying anything. Okay. Anyway. Woo. Anyway. Yeah, I hope my speech enthused you a little bit to go to data science, and that you consider it maybe as something for your future. And yeah, thank you very much for your attention.
Maybe you have some questions.
Yeah.
Thank you so much for your presentation. I would like to go back to your cancer passion.
Okay.
Why do you think we have thousands of papers with biomarker discovery focus every year, but no one results into FDA approval?
Yeah, I think that's a problem of validation, right? So that's a little bit what I was saying as well. When you produce a paper with 150 biomarkers, I mean, I'm not saying it's not true. I mean, in that experiment, it probably was true with a false discovery rate or something. But in order to find real biomarkers, you have to cross-validate over hundreds, thousands of patients, and that also differs per patient. And weak biomarkers will just fall out. So in order to have an FDA-approved biomarker, you need to do a lot of research to get through all the trials, and I think that's exactly a little bit that's missing. You know, to make that leap from a scientific sound experiment, good experiment, to make that leap from there to the clinic.
One more question here. Yes, I have a question. In your speech, you say that the main problem is to investigate the real data, high quality data, and you say that you have some methods for determination this data. But based on the complexity of, for example, this MS analysis, the influence, because on the results of MS analysis can influence even the environment around our equipment, how you choose this data. And the second one question that you describe one of your experiment when you take the data from 53 peoples and investigations weight loss, and from the point of the data science, do you think that is enough? This is a representative data only for 43 peoples?
Yeah. Okay. So the first question is that's a very good question, and that's always a problem. I think in every time when you do some sort of scientific experiment, at least in biology, you have noise, and the only way how you can do with it is you know what you're doing, but also we have lots of quality controls. So if the environment would change around the mass spectrometer, we would see that, and then we can correct for that by way of normalization. So that's
it's not the idea. My question. The idea is that, for example, even the compound of the blood of different peoples is very different.
Yeah.
And for example, in the mass spectrometer, you have some term like matrix effect. Even the matrix influence even on your target compound. By, as I understand, the main idea that in the proteomics that we analyze a high amount of substances, we use the data-dependent MS to scans, which provided by Arbitrap technology, and how you define the threshold from your correct data, because you don't know what segments give your proteomes. It's too difficult. It can recombinate. It's very high influence. How you define the correct data?
Okay. So what I think, how I would answer your question is that if there is noise in your data, and you can look at it over time. Say you have 1,000 samples, and there will be a variability because of whatever, right? And if your biological noise is within that technical noise, then there's just no way how you can extract that data. But if it's much larger than that, and then you can draw a conclusion. And the more samples you have, and the larger the biological effect, the more conclusions you can draw.
Yes,
Okay,
but
okay.
I speak not about, for example, this threshold, because, for example, the concentration of your target compound may be much lower than the concentration of trash, and you could not use, because you use not target analysis, as I understand.
We do.
You analyze the high amount of protons, high amounts of proteins, and you receive results from any of proteins which is compound in your object. Is it clear? And what is, how you find what you need to analyze using your data analysis? You can analyze the trash and receive some results, receive some correlations. How you can prove that your correlation is a real correlation?
Okay, let's do some deep dive in how proteomics works. I mean, like we try to avoid that. And you already threw a couple of terms into this, like DDA and DIA. So we do not work with DDA a lot, but actually DDA is one of the techniques that's in a way kind of more established, and they have the methods to control for the identifications, right? So if to be very specific, like you mentioned, one of the problems was that you don't know whether your quantification is valid. So here you would do the limit of quantification. It's a very typical experiment where you just change the concentration of your compound, and then you will see how this works in your signal. Just one example, right? Okay. So the second
one
will
example,
be
how you change the concentration of the hormones in the human body.
Well, you can add standards, right? So that's not very difficult.
To
so
human or to your sample?
You can add to the samples, like you would have synthetic standards in it. I mean,
it doesn't work in such, because you have your matrices, the compounds that you add in the pure water and the compounds that you receive from the human body is much different. Yes, if we're speaking about the proteins, the proteomes, because there are very many interactions.
The interaction is again, I mean, you're trying to throw a lot of problems in one bucket, right? But it's just to answer very, very short to that. I mean, it's probably like one person who would be dealing with one type of problem, right? So like, for example, Nikita, he's sitting there, he's been occupying himself for 2 months just with normalization. So normalization is comparing the intensity of the signal between the samples, right? Then you would have someone who just looks into inference of your proteins, right? So what's the probability that the peptides that you identified is actually show you that this protein is your protein, right?
Yes, but I understand. But you make a decision based on the information from several peoples. But you provide this decision to thousands of peoples, to millions of peoples from different, for example, races, from different age. From the statistic point of view,
yeah. So that's again. I mean, that's experimental design. You'll have the stages of discovery and validation. I mean, you can always throw that all together and say it doesn't work. But actually, if you go into detail and understand every problem that you're facing, there is a solution for all of them. Like, for example, statistical techniques that we've been discussing. There was also one question on the FDA approval. Nowadays, many people use frequentist techniques, right? So they will just use GLM or GE, and they would say, okay, I have statistically significant proteins, right? Do they have them? I mean, like, these techniques might not even work on thousands of samples, right? Some other people will go and add Bayesian statistics or Bayesian inference to that. They will improve their results slightly, but anyway, to the next step, you will have to validate. So one experiment that we did is ProteiQ. So we worked with athletes. We started with 10 athletes. It was our first group. We saw some potential biomarkers, and I think we identified 27 in the first run. And then you would go and validate them. So then you have a bigger trial over 6 months with more athletes. Then you'll go collect 1,000 samples. You do them, so you reduce. So you have 9. Then you go to the next stage. So for example, now we're running an even bigger trial with those 9 biomarkers. And then when you get to the stage 4, when you actually release something to the market, you still keep validating. So you get thousands and thousands. So an example that probably everyone knows is a PCA, right? So that was one of the biomarkers that was just released prematurely. And now you cannot retract it from the market, but it doesn't always work, right?
Okay, and what do I understand your solution, but now as I understand you only on the research stage. Your results does not confirm, even it doesn't validate it. It's like a theory, like a theory.
I mean,
because the results that you receive, you confirm for 10 athletes, it's something below 0. But for example, do you look at this problem, for example, from ethic point of view. When you receive the wrong positive or wrong negative results to your, for example, client.
We can talk about that after.
Yeah,
I mean,
it depends also about your experimental design, right? And what do you design? And of course, if you have 10 people, you cannot conclude that there's something for the whole world, right? But you have to take it to steps. And what you do with 10 people or 50 people, that's maybe very limited, but it already pushes in the right direction. And then you know more, and then you're going to do something else, and then some things survive and some things don't survive. And that's how you take it step by step.
And do you have in your plans to receive
maybe
the
we have
approval
more
of
questions?
FDA or EMA for your methods?
There are examples of, there are 2 FDA-approved biomarkers.
Only. Yes.
At the moment, yeah. Because, I mean, the approach is still novel. We have one question there. I mean, we'll continue this discussion.
Yes.
The question is about this MDMA stuff, MLDM stuff, multi-level disease management or...
Multi-layer disease models.
Okay. So how do you collect all the data, or are there any tools or visual tools that allow people to quickly be introduced how, for example, for cancer or whatever process is in the body, we follow from DNA, like we sequence somebody, we have DNA, then we have proteins generated. When we see how these proteins operate and what symptoms we have on the outer level, and then we connect it to some processes what were happening to the person in the past, like in few hours before or like 15 minutes. Are there any visual tools for that so that people could like synchronize, pass information about these processes between them?
Visual tools?
Yes, the tools that allow people to quickly explain what is the process in this specific case without like reading a lot of papers on some specific subjects. Yeah, the visual tools. Or any tools that help people to understand how it works from DNA level to the symptoms on the outer level, because there should be like a lot of complexity involved, and there should be some tools that display the connections between different proteins, maybe concentration like in the cell, and there's a multi-level knowledge or data that needs to be like followed forth and back. So. Like somebody needs to tackle this problem of complexity. So how do you approach this problem of complexity?
Yeah, well, for visual tools, I know for proteomics that there are visual tools. So you can look at protein interactions and these kind of things, and they're very visual and nice. If they're visual tools to combine genomics and proteomics, I actually am not aware of it. I think how the people do it now is that they just model it, but that's not visualization. So in order to visualize how the process works and get everything together, as far as I know, I'm not aware. No. Yeah, sorry.
Have some time for networking and discussions afterwards, so we'll take more questions. Maybe let's give the stage to the next presenter, and let's thank Arnoud for his presentation.